Hey, good afternoon. Today we're talking about, hey, these work here, by the way, so if you want to cool off. I'm not going to stand there, it's too noisy. We're going to talk about unusual infectious agents. These don't fit in anywhere, but they are really interesting. And some of them might not even be viruses. One of them for sure is not a virus, as you will see, but it's really, really interesting. And the first ones are called virophages, okay? And the first virophage to be discovered was called Sputnik. And this was a virophage of Mimivirus, which is another virus. So the name means virus eater. Phage, bacteriophage, the phagein is Greek, to eat. And so these virophages, as you will see, interfere. They don't actually eat other viruses, but they interfere with their multiplication, hence the name. So there is a very large virus that we talked about in this course called Mimivirus, the largest known virus with a 1.2 million base pair DNA genome. This virus, Mimivirus, infects amoeba, and the Sputnik virophage will only replicate in amoeba that have been also infected with the Mimivirus. All right, they will not replicate on their own. You can make purified preparations of Sputnik. If you try to infect an amoebal cell, it doesn't replicate. It has to be co-infecting with the Mimivirus. So it needs to replicate in the same cell, and in so doing, it reduces the yield of Mimivirus by 70%. So that's where the name virophage comes from. And this is a picture of what's going on here. Now, when Mimiviruses infect amoebae, they replicate in the cytoplasm. And they set up these factories. So MVF stands for Mimiviral Factory. So they don't even go near the nucleus. They encode everything they need, and they set up these big cytoplasmic factories. So this is a photograph, electron micrograph of an infected amoeba. Here's a viral factory right here, this dark staining material. And here are some Mimivirions here, the big ones. These are mature Mimivirions. Very unusual. They have this, these long projections coming out from the capsid, which you could just see there. And this is the factory where they're made. And you can see within the factory are there smaller particles right there. Those are the Sputnik virophages. So they're actually multiplying right within the factory set up by the Mimivirus. So they need gene products produced by the Mimivirus. So they replicate right in that factory. And they interfere with the multiplication of Mimi. We don't know how, and we don't know what specific gene products are needed. Here you can see a Mimivirus that has actually Sputniks within it. So for some reason, these have been encapsulated. Not every particle of Sputnik is encapsulated in a Mimivirus, so this doesn't appear to be obligatory. But the replication interferes with the proper assembly of Mimivirus. So here are some examples of Mimivirions that are morphologically aberrant. So here, for example, is one. The normal Mimivirus would have these nice projections. Here it's got layers and layers of what looks like membranes piled up on one side, and here you see another one like this, and here's just another. So these are effects, morphological aberrations of Mimivirus caused by a co-infection with Sputnik. All right, so this was discovered a few years ago, and it wasn't clear whether it was a general occurrence or a curiosity. And now, not too long ago, just a month or two ago, a new virophage was discovered. It's called Mimivirus. MA stands for maverick. And maverick is a kind of transposon, a DNA transposon. These virophages appear to have homology with DNA transposons. This is a virophage of a virus that infects this little guy up on the upper right. This is Cafeteria renbergensis. This is a marine phagotropic flagellate. So it's a eukaryotic organism. It's a protist. It eats bacteria. So phagotropic means it gets its nutrition by phagocytosing bacteria. And the people who discovered this said we have to call it cafeteria because it eats a lot. So they call it Cafeteria renbergensis, all right? So it lives in the oceans. In fact, it may be the most abundant predator in the oceans, this guy right here. So there is a virus that infects it, and this virus is called mRNA. So I'm sorry, mRNA is a virophage of a virus that infects these cafeteria guys. Now, this virophage has been discovered in a lake up in the Antarctic. Arctic. The Arctic is on the North Pole, right? Arctic. The Arctic Circle. Organic lake is all the way up as far as you can go north without reaching the North Pole. There's a lake up there. I'm sorry, it's Antarctica. It's below. I'm thinking of another viral story up in the Arctic. The organic lake is in Antarctica. It is a hypersaline lake down there, which is frozen most of the year, but covered with ice, but at certain times of the year it thaws out. And it has microbial life mainly in it. And they found this virophage in algae that are infected with a ficodnavirus. So this is a separate story from MA virus. MA virus is a virophage of a giant virus that infects these guys. The organic-like virophage infects algae that are infected with ficodnaviruses. So ficodnaviruses are large DNA viruses that infect algae. And down in these lakes in Antarctica, they find this virophage. They also find this virophage in other aquatic environments as well. Now that we have a few virophage sequences, you can start saying, where else do we find these? They found it in a hypersaline lagoon in the ocean near the Galapagos Island, in the Delaware Bay near New Jersey, and also a freshwater lake in Panama. So there have been sequencing projects at all these sites, and all they had to do was go and mine the sequence information, and they found evidence for virophages. And these virophages, both MA virus, Ma virus, and the organic lake virophage, they all share a couple of genes with the virus that they prey on. And they have other genes as well, and they have homology, so you can search for those in databases. So one of the ideas for what these are doing is that they may be exchanging genes among viruses. They may be responsible for gene exchange by recombination, and that encourages diversity. They may also have an impact on the ecology of the ocean. An example of this was shown in the study of Antarctic organic lake. So they assume in this study that the virophage inhibits the production of ficodnaviruses. They haven't actually done the experiment, but they assume it does, just like Sputnik inhibits Mimivirus. And if that's the case, they did a modeling study of the microbial community in this lake. This microbial community is mainly algae. And what happens in this lake when there is sun available, which is not all year, the algae bloom, and then the viruses kill the algae and they release carbon. So they do a carbon cycle. And there are alternate cycles of blooming and virus killing down in the lake. Now, if you take the virophage and you model that into the equation for the oscillation of the algae populations, what you see is that the virophage lets the algae live longer. So they have more extensive blooms, and they're less susceptible to killing by the ficodnavirus. So this is all modeling, mathematical modeling, but the idea would be that these virophages could regulate the carbon cycles, especially in places like Antarctic when you don't have sun all the time and it's very difficult to make an algal bloom. These viruses may have evolved to help the algae grow. So that's a very interesting idea, and it may be that since we find these in other aquatic environments, they're also involved in cycles there as well, because we know that viruses play important roles in many of the cycles. For example, the carbon cycle in the ocean. So these may be modulators of that. So they're very interesting. So they're relatively new, and by this time next year we'll have even more information on them. All right, so that's virophages. Yes?
Do you expect virophages to be only in water?
Good question. I mean, maybe you have virophages in you. Maybe we all do. Why not? Now that we have the sequence, we can look for signatures in all the databases. But I don't see why there wouldn't be virophages of other viruses as well. Sure. I mean, so far they've only been discovered in these big DNA viruses. So one idea is that maybe those are the only ones that can sustain them, but we'll see. Yeah?
Are they encoded in the virus itself or in the host cell?
They have their own genome. So the virophage has its own genome and encodes, it's about 20 kb in size, and it has some genes that have high homology with the virus. Okay, and then a lot of the other genes are unique. So it's got its own genome, but it just can't replicate without co-infecting a cell that also have the other virus in it. Okay. So this is new this year. We didn't have this in last year's class. So just since 1 year, all this has been learned. Okay, so that's virophage. All right, the rest of the time we're going to talk about some other very unusual agents, and they really start to answer these questions. What is the minimum genome size needed to sustain an infectious agent? It's the smallest genome. And could an infectious agent exist without any genome? So the agents we'll talk about are going to address these two questions. And the ones we'll talk about are viroids, satellites, and prions. And these don't fit into our taxonomic scheme for viruses. As you will see, we call them subviral agents. Viroids are important pathogens of plants. Agriculturally extremely important, as you will see. Some satellites are associated with human disease, and prions cause very severe dementias in people. Okay, so those are the three we will talk about. Viroids consist only of RNA. There's no protein in a viroid. So, we have been talking in this course about how a capsid's job is to protect the genome, but these don't have a capsid. They exist as pure nucleic acid. Yet they're still able to get from host to host without a capsid. They don't need receptors at all. I just checked the subviral RNA database this morning. It's got 2,883 sequences for viroids and related RNA molecules. So it went up about 13 from, there's a number, your handout is the old number, so it went up a bit since last year. So there it is. The first viroid was discovered in 1967, potato spindle tuber viroid. This is how we abbreviate these. VD is the viroid abbreviation. And it's the prototype for all the viroids we're going to talk about. Smallest known nucleic acid-based agents, 350 nucleotides in the case of PSTVD. Some of these viroids don't do anything. They seem to infect and replicate in the host without any harm. Others have economic consequences, particularly in crop plants. If you have a crop, you don't want certain viroids infecting it. So viroids are just naked RNA, and not only are they naked, but they don't encode any proteins. So really, this is the minimal genome. Yet these can replicate, as you will see. The RNA is small, typically circular, single-stranded RNA, highly structured, though. When this is introduced into plants, these RNAs replicate. So these used to be called subviral agents, but now we realize they're not viruses, and they're not subviruses. So we have classified them into two families, Popsiviroidae, Pops P. viroidae. Okay? These replicate in the nucleus of the plant, and Avison viroidae replicate in chloroplasts, as you will see. Okay? So these are some of my favorite viroids. The kadang coconut viroid. It's because the name is good. It causes a lethal disease of coconut plants. And if you ever see a palm tree like this one where the leaves are brown, likely that it has a viroid infection. And this is affecting the yield of coconuts. So if you like pina coladas, it's not a good thing. The hop latent viroid is a viroid that affects the hop plant. What's hop used for? Beer. Yes. So you're happy because it doesn't do anything to the plant and it's okay. And then there's the apple scar skin viroid, which just makes these cosmetic alterations on the surface of your apple. You ever see an apple like this, anyone? Would you eat it if you saw this? No? Would you eat it? No, it's not going to make you sick. None of these viroids infect people, no. So you could eat it. But in the supermarket, they're likely to be the ones left behind because people think there's something wrong with them. But if you ever see this, it's fine. You can go ahead and eat it. Right? But if you get sick, don't call me, right? Because it's not the viroid. Okay, so they don't encode proteins or mRNA, right? No capsid, no protective coat, no protein. It's just an RNA molecule. Circular, single-stranded, smallest is 246 nucleotides, the biggest 399. And the RNA is extensively base paired, as you will see. Looks actually like a rod when you look at these under EM. They don't look like circles. They look like rods. And some of the viroids have an activity called ribozyme activity. Does everyone know what a ribozyme is? Anyone don't? So ribozymes are self-cleaving RNAs discovered some years ago. And these are essential for the replication of these viroids, as you will see. So the ribozyme is not in all viroids, but it is in many, and we'll see what that does. How do these differ from viruses besides not encoding any proteins or not having a capsid? Well, the viroids are parasites of the transcription machinery, as you will see. All they have to do is replicate their RNA genome. They don't have to make any proteins. So they want the transcription machinery. Whereas viruses, as we know, all viruses are parasites of the translation machinery. No virus can translate its messages on its own. Some viruses can make messages, right, as we've learned. Not all, but some. So the ones that can't, they're parasites of the transcription machinery. But in general, if you want to make a generalization, all viruses need to parasitize translation, but the viroids need to parasitize the transcription machinery. So here is a picture of potato spindle tuber viroid, RNA. As you see, it's a circular RNA, and it base pairs and folds into a rod. So if you denatured this, it would be a circle. And it's been extensively studied as an agricultural pathogen. It has very distinct regions. It has left and right termini, which form a loop. It has a region that's been involved in pathogenesis. If you mutate this region, if you change the bases, you influence the pathogenicity. So this viroid, when put into potatoes, makes them unedible, basically. You can't sell them. So that's what we mean by pathogenesis. And then there's a central conserved region as well. So how do they replicate? Here's what happens. When the viroid gets into a cell, it is copied by host RNA polymerase II, the same host enzyme that would make messenger RNA from our DNA genome. So it makes concatemers, which means it has multiple unit lengths of the viroid within a longer molecule. And those concatemers are then cleaved to form unit length viroids, in some cases by the ribozyme that's actually encoded in the viroid RNA. So again, the ribozyme is a sequence. It's a structured RNA that has the ability to cleave itself. All right, so these are embedded in many viroids. So you can go from having a concatemeric replicated state to individual viroid genomes. These were discovered, by the way, in 1981 and resulted in Nobel Prizes to 2 individuals. And they may be relics of the RNA world when there wasn't protein around, and this was one of the enzyme activities that was present. Yes?
So viroids use host RNA polymerase to make RNA from RNA.
Right. They used host RNA polymerase II. They encode nothing of their own. So they can't, you can't go wrong with a viroid in saying it has to have everything given to it by the host. So it's made by host polymerase II, and then in some viroids, the ribozyme cleaves the concatemeric molecules. So one of the ribozymes that are present in viroids is called the hammerhead ribozyme. There are different kinds of ribozymes. This is an autocatalytic self-cleaving RNA. It's a rather short sequence. You can transport it to almost any RNA, and it will cleave itself in a different surrounding. And as I said, it uses this ribozyme to cleave the concatemeric structures that are made during replication. Now, not all viroids use the ribozyme. Not all of them have it. And then those that don't have it appear to use nuclear enzymes to cleave their concatemers as well, instead of the ribozyme. So 2 distinct evolutionary pathways. So here's the replication scheme of the members of the POS P viroidae in a plant cell. Now, these RNAs get into cells most likely by mechanical damage, farm machinery breaking the leaves or the stems, and that introduces the ribozyme because there's no vectors, as we know, and there are no receptors needed. The RNAs are brought into the nucleus. Here's the ribozyme, the circular blue molecule, that's the ribozyme. Host Pol2 copies it and makes concatemers, so now we have two genome units here, but they're actually longer. Now this is labeled minus, but plus and minus really don't mean the same thing as they do when we talk about messenger RNA. Remember, messenger RNA we always define as the plus strand, but these don't encode any proteins. So there can't be a plus and a minus. It's just a convention so that you can think about how these replicate. So I just don't want you to think that the plus strand is a messenger RNA. So the initial concatemers are copied again to make the plus strand, probably also by Pol2, because the virus can't do it. These are imported into the nucleolus then, where the concatemers are then cleaved by a nuclear nuclease, most likely. So there's no ribozyme in these particular viroids. They're then exported out of the nucleus, and then they can move to the next cell, the way material moves among plant cells. All right, so the key is everything's done by the host. In this case, copying of the genome and the cleavage. And that's how these replicate, and that's it. You don't need to make any proteins. You just take advantage of the host. So you can see that if this is possible, then it evolves, and that's what happens in the case of ribozymes. Here's the other family of viroids. This is what happens in the case of viroids. In the other family of viroids, these are the ones that have hammerhead ribozymes. That's why we're drawing it like this. Again, these get into cells. They go into the chloroplast, not the nucleus as in the other case. They go in the chloroplast. They are copied, again, by the cellular RNA polymerase II. And in this case, the concatemers are cut by the ribozyme activity of the viroid itself. And then these are copied again to form plus strands, which then get exported from the chloroplast and spread from cell to cell. So you just need to have one viroid get into a plant, and once it's there, it will spread systemically throughout the whole plant from cell to cell. All right, so this is different because you make initially unit-length genomes, and then you copy them again. Whereas the other strategy, you make concatemers, copy them, and then cut them. Again, all done by host RNA polymerase. Yeah? Not all viroids code for ribozymes. Both strands, and both strands. In this case, it's on both strands, yeah. Because you make concatemers and they get cleaved, and then you make another concatemer of the other strand and they cleave themselves also, yeah. So this is an example of peach latent mosaic viroid. And just to show you where the ribozyme sequences are located, here's the entire viroid. You can see it's extensively folded. Here are the cut sites, here and here, for the two different strands. So here's the ribozyme for the top strand, and here's the ribozyme for the bottom strand. So just to show you how these fold, in the cutting, this folded RNA has catalytic activity, and it cuts either here or right here. And that allows the concatemers to become individual genomes. So where did they come from? So they've probably been around before agriculture. In the 20th century, we probably introduced them into our crops ourselves. When we were breeding modern crops, you start with wild plants, and probably some of the wild plants had viroids in them, and we transferred them to our modern crops. We do a lot of monoculture. We used extremely hybridized plants. Most of the crops of different sorts in the world all come from the same plants made by the same companies. They're genetically identical, and if they have a viroid, everyone gets the viroid. And these plant lines are global, so viroids are global as well. So as I said, now they're probably transmitted by farm machinery. If the viroid is in part of of a farmer's crop, and that farmer uses mechanical equipment to plow, that mechanical equipment will transfer it to another plant. How do they cause disease? So some viroids don't. They just replicate and the plant is fine, but others do compromise the plant. They can ruin a whole crop, depending on the plant in question. But how can they cause disease without encoding any proteins? So we think this all has to do with small interfering RNAs. So we believe that there are small RNAs derived from each viroid RNA. These are produced by the silencing machinery of the host, the Argonaut proteins and all of those. And these RNAs probably silence host genes, and that causes the pathogenicity. In fact, symptom development correlates with the production of these RNAs. If you inhibit their production, you have less symptoms. And we think that some of these small RNAs are actually located in that pathogenicity determining region that I showed you on an earlier viroid. So as I said before, if you make alterations, mutations in that region, it affects the pathogenicity of the viroid. So we think this contains the RNA sequences that will eventually be chopped down to 21 nucleotide interfering RNAs, which will then interfere with the host. Okay? So the symptoms are a consequence of interfering. But again, not all viroids do this. Only certain ones have done it. It's not clear why that would be. Okay, so that's viroids. That's our first unusual class of infectious agents. Yes? How do they get in? Mechanical damage. Because there's no insect vector, there's no receptor, it's not brought in by a bacterium or any sort, so it's probably mechanical damage. Somehow contaminates farm equipment, shovels, tractors, and that's how it gets in. Yeah. All right, the next unusual infectious agent is the satellite. And these are, again, small RNAs. They're single-stranded. They're a bit bigger than the viroids, 500 to 2 kb. They lack genes that they need to replicate. These guys do encode some proteins. They can code 1 or 2 proteins, but they don't have all they need to replicate. So they depend on a helper virus. They can only replicate in cells which are also infected by the helper virus. So those are satellites. These viruses, well, these satellites form particles on their own. And in fact, in most cases, their genomes, the proteins that they encode, are part of the capsid, either the whole capsid or part of it. So the replication proteins, the proteins they require from the helper virus, help them to replicate. They are not derived from the helper virus. They're completely unrelated. They have no homology with the helper. They just need functions provided by the helper virus because they don't encode enough proteins on their own. So they're different from viroids because they do encode proteins, but they're still dependent on a host. So on a virus. The viroids needed the host to replicate. The satellites need another virus. Most of the ones we know of are also associated with plant viruses. And in a plant that's infected with a satellite plus helper, the disease is different than if the plant were just infected with the helper virus by itself. So the helper can replicate on its own. It doesn't need the satellite. The disease symptoms are distinct. So here are some examples of satellites of bacteriophages in animal viruses. So an E. coli bacteriophage called P4 is a satellite. It requires P2 as a helper. It cannot replicate on its own. Doesn't have enough proteins. So it only will replicate in E. coli that are also infected with P2. The eukaryotic animal viruses, adeno-associated viruses, these are members of the Parvoviridae. We've talked about these a bit, especially with respect to DNA replication. These require adenovirus or herpes viruses as helpers. So again, they are defective in the sense that they don't have enough genes to replicate. They need another helper virus. But again, they have no homology with adenovirus or helper, nor does P4 have homology with P2. You could actually think that the virophages we talked about are like satellites, right? Because they form distinct particles They have some genes, but not enough to replicate. They do need a helper virus. The differences that I can see is that they're much bigger than the typical satellites, which may not mean anything. And they also inhibit the production of the helper. Now, it may be that there are some real satellites out there that also inhibit the helper virus. I'm not aware of them at the moment. But maybe virophages are, in fact, satellites that have just evolved to be bigger. So we'll see. So it could be that at some point in the future, we'll talk about satellites and we'll include the virophages in them. Okay, this is an example of a human satellite. It's called Hepatitis Delta virus. Now, this is a sort of hybrid between a viroid and a satellite, as you will see. The helper virus for Delta, it's a satellite, it requires a helper, is Hepatitis B virus. So this has to co-infect the same cell as Hep B in order to replicate, as you will see. Now, it used to be that when this Delta was first discovered, it was thought that it made hepatitis caused by Hep B more serious in people. That doesn't seem to be the case anymore. There have been a lot more epidemiological studies, the results of which suggest that this is not the case. But it's out there, and in fact, there are about 18 million people globally who are infected with hepatitis delta virus. That's about 5% of the 350 million carriers of hepatitis B. So a lot of people are infected with Hep B globally. And here's the distribution of Hep delta globally. For a time, it was also invading Europe, but the numbers in Europe now seem to be declining. And prevalence is rising in the Asia-Pacific region. So 18 million people have this satellite. What is it? It's a 1 point... Yes?
Going back to the map,
Yep.
does it have any correlation with the Hep B prevalence as well?
Yeah, it does. But it's not everywhere there is Hep B. But it can't be. It cannot be anywhere where there is no Hep B.
So the carriers who have HDV has Hep B?
Yes, absolutely. Nobody with HDV doesn't have Hep B. You have to have Hep B. So Delta carriers are a subset of those who have Hep B. And that's 18 million out of the 350 million with HBV. All right, the genome is a 1.7 kb RNA. Again, it's single-stranded, it's circular, like the viroids, and it folds upon itself to make a rod-like structure, so it's highly base paired. It also has a ribozyme, like some of the viroids do. It encodes one protein, which is called the hepatitis delta antigen, and as we'll see, this protein encapsulates the genome, or it participates in the encapsulation. Forms part of the coat of the virus. And when this virus comes out of a cell that's infected with Hep B, so again, it has to multiply in a Hep B infected cell, it acquires the coat, the envelope proteins of Hep B. So the virus particle is the Hep B surface antigens and the delta antigen. We have a picture of this to make that clear. All right, so this is a schematic of the genome of delta. Again, it's a circular single-stranded RNA, but it's over 70% base paired, so it forms a rod-like structure. And we have a genome and an antigenome. So this is actually a better nomenclature rather than plus minus, which we use, well, not we, but those people who study viroids call them plus and minus, but genome and antigenome is a bit better. Although this satellite does encode an mRNA. It contains one mRNA, which of course is produced from the antigenome, and it encodes delta antigen. So here's the open reading frame, and there are two versions of this protein produced, a large and a small version. And it also has a self-cleavage site. It's the red circle here. So again, this is a ribozyme. It can cleave itself, and that is because this replicates by making concatemers just like the viroids do, and those are cut by the ribozyme. So here's a schematic of the replication, just to give you a sense for how this is happening. It's very much like the viroids, except that a messenger RNA is made. So here's your incoming genome. It's copied by host polymerase II to make a concatemer. You can see you have a 1 genome length version here and then a little bit more. These red dots are the ribozyme cleavage sites. So this cleavage here, for example, is giving you an mRNA, which encodes for delta antigen. The replication continues. You make longer molecules, and you eventually get full-length plus strands out of these. And then you copy those again to make minus strands. So the point here is that an mRNA is produced, and there is also production of long concatemers, which are resolved by cleavage, self-cleavage, at these ribozyme positions. So that gives you genomes and antigenomes, and this mRNA, which encodes for delta antigen. So again, this is done by the host polymerase II, because this is an RNA. It has to be copied by an enzyme. There's no enzyme encoded in the genome that can copy it. So this is done by host pol II. So for a long time, it wasn't understood how this genome would be copied, because I have spoken many times in this course about how viral RNAs can't be copied by host RNAs, right? By host polymerases. So this is an exception. The delta genome can be copied, although it's not a virus, of course, it's a satellite. And here is how we think the virion is put together. This is a hepatitis B virion here on the right. It has an icosahedral capsid and an envelope around it, which has the viral glycoproteins in it. And here is delta virion. It's smaller. So in this picture, the delta genome is in the middle. Here it's coiled up because it's base paired, rod-like structure. It's coated with small delta antigen along its length. Then there's a large delta at either end, which is believed to hold it to the glycoprotein. So these delta particles bud out of the host cell, and they acquire the envelope of hepatitis B virus. So they have glycoproteins of hep B with a delta genome inside. So these can go on and infect the same cells that would be infected by hepatitis B virus. But if those cells are not infected with hep B, the delta genome will not make particles. It will replicate in that cell, but it can't make new particles. So it's completely dependent on hep B for this envelope and glycoproteins. Okay? All right, so that takes care of our unusual agents with genomes. And that brings us to infectious agents that don't have any genomes, that consist just of proteins. And you may ask, why are we talking about this in a virology course? And because they don't have a genome, so they're not viruses, they're just pure proteins, they're infectious proteins. And, well, I think they're very interesting because they have a transmission pattern which is very virus-like. But maybe more importantly, when the existence of these agents was first proposed by Stanley Prusiner, for which he got the Nobel Prize eventually, no one would listen to him except the virologists. And so historically we have a warm feeling in our heart for prions. And that's why they're in virology textbooks. Nobody knows where else to put them. But I think they're fascinating. So these are prions, and you have heard many of the diseases caused by them. Mad cow disease, bovine spongiform encephalopathy, Creutzfeldt-Jakob, Scrapie Kuru chronic wasting disease of elk and deer. In fact, this is starting to spread in deer in the U.S. And they tell you if you go out hunting deer, you should watch out for the spinal cord and brain. Stay away from it because that's where these infectious proteins reside. Because you might be infected. There's no incidence of going from a deer to a human yet, but who knows? Anyway, so Prusiner got the Nobel Prize for this. We'll talk about what he did. So these proteins called diseases that are called transmissible spongiform encephalopathies. All right, so prions are infectious agents without genomes. And this name means a fatal neurodegenerative disorder of mammals, just mammals we're talking about here. And thousands of humans come down with this every year. About 1% of these cases arise by infection. We'll talk about how the others arise as well. For example, by 2012, 120 people had contracted Creutzfeldt-Jakob disease by eating meat from animals with BSE. These were the mad cows in England, and we'll talk about that in a little bit more detail. So it's a transmissible. It can be transmissible. About 1% of the infections are transmissible. Spongiform refers to the pathology in the brain, which we'll see, and encephalopathy means disease of the brain. So here are some of the TSE. I'm going to abbreviate that as TSE, transmissible spongiform encephalopathy. These are some of the diseases of animals that we know of. There's BSE, chronic wasting disease of deer, elk, and moose. Exotic ungulate encephalopathy. Feline spongiform encephalopathy. Your cat can get mad cat disease, I suppose. Scrapie in sheep and goats. And transmissible mink encephalopathy. So that's in animals. And then in humans, we have Creutzfeldt-Jakob, fatal familial insomnia, Gerstmann-Straussler syndrome, Kuru, and variant Creutzfeldt-Jakob. So variant CJ is the disease that you get from eating infected cow meat. It's slightly different from the original Creutzfeldt-Jakob. Okay, so let's start with a little history. Scrapie is a disease that's been around for a while. It's been recognized in European sheep for over 250 years. It's the first TSE that was recognized. In the sheep, the disease gets its name for the fact that infected sheep rub themselves or scrape themselves on fences. So the farmers called it Scrapie. And the sheep get motor disturbances, trembling, and in Canada the disease is called tremblement du mouton. Right? It's not? I didn't pronounce it right, yes, of course. That's what the disease is called. Paralysis, weight loss, death within 4 to 6 weeks. And in the UK, it's in 1% of the sheep. 1% of the sheep each year get it, so it's still around. We haven't been able to do anything about it. So that's a sheep with Scrapie, and they, besides the neurological symptoms, they rub themselves, so they scrape all their fur off, and they have large patches of skin showing. He doesn't look very happy, right? Yeah. So he's going to die within 2 months. The farmers were doing the epidemiology here. They found that the animals that were infected could transmit the infection to another herd. They could, if they were physically present, they would transmit the infection. So they made the first important epidemiological observation. And then in 1939, it was found that if you took the brain of a sheep with Scrapie and you ground it up and you pass it through a filter, the infectivity goes through filters that normally passes only viruses, which would have suggested that it was a virus, except that, and initially that's what it was thought to be, but then it was, as it was studied, this infectivity or this agent was found to have very unusual properties. It was highly resistant to UV, ionizing radiation, and formaldehyde, things that inactivate viruses. And then when people were looking for nucleic acids, they didn't find any. So they would take these preparations that infected sheep, and they couldn't find any nucleic acid. And here's a result of an experiment where we're looking at the molecular weight of nucleic acids of various agents and the dose of radiation that's needed to inactivate the genome. So low doses to high doses here. So the viruses with big genomes, so herpes and vaccinia, it doesn't take a lot of radiation to inactivate their genomes because they have a big target. This is all about target size. The bigger the target, the easier you are to hit. As the genomes get smaller, you need more radiation to knock out the infectivity. So you can see here polyoma, the small DNA viruses, some of the small RNA viruses, they're quite resistant to irradiation. But look at the Scrapie agent here. It's more resistant to radiation than any known viral genome. So this is one of the first clues that these were unusual, that they might not have nucleic acid because they were more resistant to irradiation. There's a huge amount of radiation needed to inactivate the agent than any other known virus. Now the disease that are caused by these agents, remember we said it was spongiform transmissible encephalopathy. So a spongiform means that the infected brain is literally full of sponge-like holes. And this is accompanied by psychomotor dysfunction, and the particular symptoms depend on what part of the brain is infected. Each disease that we talked about in that slide of listing all the diseases has its particular symptoms and pathology. And here's a spongiform pathology. This is a section of brain from an infected sheep with Scrapie. And although I'm not showing you a normal control, you shouldn't have all these holes in this section. So that's the spongiform appearance caused by this agent. And so the symptoms are in part resulting from this damage. Yes? Could you make a vaccine against this? Well, as you will see, it's a protein of yours that's causing this disease. It's not a foreign protein. All right? Well, so we're going to get to that now. But it's your protein, so you can't immunize against yourself. So you might be able to if you could induce somehow antibodies against the specific conformation of the protein that is pathogenic. But that's kind of difficult. So that's the problem. Okay, so all the TSEs, the human and the animals, have very similar histochemical abnormalities. There are plasma membrane defects. You get vacuolization of various cell types, loss of neurons, the spongiform appearance, which we just saw, and you get accumulation of this protein, gliofibrillary acidic protein, in clumps. And finally, they're all characterized by amyloidosis, and that is formation of fibrils of amyloid protein. So whenever proteins accumulate and form fibrils, call it amyloid. So this is one of a number of diseases that do this. And along with the spongiform appearance, it's a characteristic marker. So you can detect the disease by injecting organ homogenates from one species into another. And then what happens is that you take a sheep with Scrapie, you can make a brain extract, inject it into another animal, and then you eventually get symptoms of the disease, difficulty walking, dementia, and death. But the incubation period is very long. It can be months to years. So if you're studying this, you have to be very patient. And when you inject these extracts into animals, it accumulates in very specific places and then eventually gets into the CNS. So as you'll see, you can actually acquire these TSEs by eating infected meat. So it can go from your intestine, eventually making its way to the CNS. And as I said, in the CNS you have astrocytosis, vacuolization, which is the spongiform encephalopathy, and loss of neurons. There's no antibody. There's no immune response. There's no inflammation, which you would get early in infection with a virus, the cytokines being produced. There's no antibody produced. There's no cellular response, because this, as you will see, is our protein, which is causing this problem. And that's why we don't get an antibody response. So this is difficult because it makes it harder to detect it. You know, with most infections, we can use antibodies to detect the infection, but you can't do that here. So the problem is that with most human TSEs, you don't know you have it until you have symptoms, the neurological symptoms, and then it's too late to do anything. Not that we can do anything, because they're all invariably fatal. But at some point, we theoretically will have a way to ameliorate the symptoms, but we can't detect it right now before clinical symptoms. So we have no diagnostic tools to determine who's carrying the agent. People are working very hard at this, and there are some good leads, but not yet. And of course, it's not treatable. But as I said, I think someday we will have treatments, in which case we need diagnostics. And it's 100% fatal. So in 1967, after all these data were accumulating, Griffiths suggested that the agents of TSEs were in fact protein. So he looked at all the data, the ionization radiation data, the purification, all of this, and he decided there must not be any nucleic acid in these. And then Prusiner began working on these. He eventually purified the protein that would transfer Scrapie from a sheep to another animal. So he started by making brain extracts from Scrapie sheep, passing them on to other animals, and then he would purify the extract and ask what part of the extract is causing the disease. And he eventually got it down to a single protein. It took a long time because the incubation time is up to a year for this. And he ended up calling it a prion, proteinaceous and infectious particle, because he found that it was just a single protein. So he identified the protein, got some sequence, and then he could identify the gene. It's encoded by the Prion P gene. So it's a normal gene in us that encodes this protein, which under certain conditions can become a transmissible agent of TSEs. And this gene is essential for the pathogenesis. If you don't have this gene, you will not get a TSE. You can delete it from mice, and the mice are fine. I don't know if you can delete it from people, though. I haven't done that, but it doesn't seem to be essential. Right? So maybe someday therapy will consist of antagonizing the function of the protein. So our current view is that a prion is a conformational isoform of a normal protein. So the normal protein we call PRPC. Prion protein, C means cellular. It's normally on the outer surface of neurons. This protein is anchored by a GPI anchor. So it has some normal function. The abnormal version, we call an abnormal conformer. If you take that abnormal conformer and put it into a host, it will cause the normal host's prion protein to be changed into a pathogenic version. So let's say you have sheep and you purify the prion protein from them. That protein is what we call an abnormal conformer. Its structure is different, and I'll show you what we think it is in a moment. You take that and you'll put it into a new sheep. The abnormal protein catalyzes the conversion of the sheep's normal protein into an abnormal form. All right, so that's what we mean. The abnormal conformer causes conversion of normal PRPC into the pathogenic form, and we call that PRPSC. So SC means Scrapie. So prion protein cellular is the normal version, and PRPSC is the pathogenic version. If you take a normal version, if you take PRPC and inject it into someone, it doesn't cause disease. But if you take PRPSC, which you can only get from infected animals, and inject that into another animal, you get a TSE. So this PRPSC in an animal with disease accumulates mainly in the brain. No matter whether you ingest it or inject it, it gets to the brain and accumulates there. Yes?
Is there a threshold value of the presence of the PRPSC, or is there, I can't imagine it's just being one and causing some widespread transmission of PRPSC.
You can't imagine that one abnormal can cause the... Well, we have some models coming up for that to try and figure, because that's a puzzle. Yeah, but you get a lot of accumulation of the PRPSC. Eventually, that's why you have disease, because you have pathological levels. So disease doesn't result from just a few aberrant proteins. You have to have a lot of them. Right? All right, so these aggregates accumulate in the brain. They make amyloid, which are the fibrils of protein. And that's prion protein, and you get these spongiform holes. And this protein, this disease version, PRPSC, is also protease resistant. And that was an important finding for purifying it, as you'll see. So this is a staining of a section of brain from an animal with a TSE. And the antibody used to stain this is an antibody against prion protein. And these are amyloid plaques, so these contain fibrils of prion protein, and you can see they're very discrete depositions of these in the brain. So that's what we mean by amyloid plaques. If you look at higher magnifications, you can see the highly fibrillar nature of these plaques, which is the accumulated prion protein. So normally, your prion protein that you make, we all make, it's called PRPC. We know the sequence of it. It's GPI linked, and again, it's on the outer membrane of neurons. If you treat this with proteinase K, which is a kind of protease, will completely digest this protein. PRPC is completely sensitive. But what Prusiner found was if you take the PRPSC from, say, sheep who have Scrapie and digest them with proteinase K, all it does is clip off the N-terminus. The rest of the protein is highly resistant to proteinase K digestion. It's the same protein. It's exactly the same sequence. Yet somehow it is folded in a way that it's resistant to cleavage with proteinase K. Okay, so we know the structure of PRPC, and that's shown here. This has been solved by X-ray crystallography. We don't know the structure of the SC conformation. It's a very difficult protein to work with. But the idea is that somehow going from PRPC to SC, you have a conformational change that makes it resistant to protease digestion. Part of the change involves making a lot of beta sheets, which is shown by this here. The protein has a high content of beta sheet, and that's probably part of the reason why it's resistant. So this shows that it has conformational difference. PRPSC is very different from PRPC. Yeah? We know the sequence of S. It's the same as C. It's exactly the same protein, but we don't know the exact conformation. Yeah, so that's what I want you to remember, that PRPC and SC have exactly the same amino acid sequence, but one causes disease and the other doesn't. So it must be that there has to be something inherent in this protein, and probably this conformational difference is part of it. So what causes the disease, the key to understanding what causes it is going to be to understand what catalyzes this transition. We just don't know that yet. Now, in mice, you can do very nice genetics. You can knock out the gene encoding the prion protein. You can knock out the gene encoding PRNP, the normal cellular homolog. These mice are fine, as I said, and if you infect them with prions, they are totally resistant to infection. They never develop TSEs. So that shows you that you need your own copy of the prion gene in order to develop the TSE. And that's partly where this idea that the pathogenic protein is inducing your normal protein to change into an aberrant form came from, because you need your prion gene in order to develop the disease. So sheep spontaneously seem to come down with Scrapie, and it may arise as a genetic mutation in them. So there may be spontaneous single amino acid changes in the prion protein which make it misfold and become pathogenic. So those are rare mutations. Or you can acquire these TSEs by infection. So sheep can get it from each other. So those are two ways that these appear, by infection or by a rare mutation that causes the protein to misfold. So the ideal proof of the Prion hypothesis would be to make conformationally altered PRPSC in vitro and then infect an animal. And people are working on that. It's not quite possible yet to do that, but at some point it will be done. But there's a lot of very strong evidence for this entire hypothesis. So how does a normal protein make an abnormal version of a protein in you? I'm sorry, how does the Scrapie protein, the abnormal protein, cause conformational change in your normal protein? So there are many hypotheses. These are just two. Here's one on top where here's our normal PRPC. And this PRPC theoretically can change into the pathogenic conformation. Here you see that in this model it's not happening. There's no alteration between the two states. So in you just have the cellular conformation. But if you encounter some abnormal protein, if you eat infected meat, for example, then that abnormal protein can catalyze the conformation of your normal protein to make abnormal ones. And then those can go on and catalyze the conversion of even more. So then you can see that this cycle would amplify itself, and eventually your normal protein, lots of your normal protein would be converted. That's the refolding model. So the idea is that the Scrapie version of the protein causes the normal cellular version to refold. The seeding model, there is some equilibrium between the normal and the pathogenic forms of the protein. So if you have some abnormal protein forming, it would very, very slowly convert to multimeric forms. And then as you add more abnormal protein, these would eventually form the amyloid fibers leading to plaques. So this says that there is extremely slow conversion, and in some of us that leads to disease. It's very rare, and that occurs by this misfolding. Yes?
Is it just coincidence that you've only found this in the brain? Or is there a reason that this sort of thing couldn't happen in different organs?
Well, this happens to be a brain-specific protein. It theoretically, it could happen somewhere else, sure. And maybe there are diseases that we don't know about. Yeah, sure. There's nothing brain-specific about the mechanism, no. Okay, there is a very nice story about human TSE, which is actually the first TSE to be discovered. It's called Kuru. It's a fatal TSE in the 4 people of New Guinea with a 30-year incubation period. An American epidemiologist, Carlton Gajdusek, studied this disease, and he found that it largely attacks women and children, and that's because when people die in the 4 people, they're the ones who eat the brain of the deceased person. The men don't get to eat the brain, just the women and kids, and they develop Kuru. So he studied this and he said, you're having Kuru because of this, so you have to stop the cannibalism, and they did, and the disease is now gone. So this was the first human TSE known, and it came about, it was perpetuated in this people because of cannibalism. How it was introduced, well, probably years ago, someone had a sporadic version of the disease where they just converted to the pathogenic form by maybe a mutation or some other environmental insult, and someone ate that person's brain when they died, and that started the epidemic in this tribe. So human TSEs can be infectious or transmissible like Kuru. You can have spread of these diseases by transplantation of infected corneas, by giving people hormones that are contaminated, by transfusions. This has been documented to occur among people. Of course, BSE you get by feeding ground-up infected animals to cattle, and that ended up causing a variant type of disease in humans called variant CJD, which is you get by eating BSE beef. So basically, forced cannibalism spreads BSE. If we had an epidemic of BSE in the UK, there's a form of cannibalism, and this is because we fed the cows chopped up other animals. So they got a homogenized high-protein diet. The cows normally don't eat meat, right? They're out there eating grass, but farmers want them to grow quicker, so they give them protein. And in the UK, they were giving them ground-up carcasses of other animals. And some of those animals had died of Scrapie without anyone knowing it, so the cows got a form of a TSE called BSE, or mad cow disease. A lot of similar symptoms: dementia, ataxia, spongiform encephalopathy. So this was recognized in the UK. They stopped feeding this rendered stuff to the cows, and they stopped getting BSE. But a lot of people ate BSE contaminated beef, and they in turn got a variant TSE called variant CJD. So this is a map of the epidemic in the UK. The onset of bovine mad cow disease, BSE, in about 1987. You can see this is when the first cases were discovered, and then they banned the use of this ground-up feed in cows, but it was too late. This has a very long incubation period, so you can see the disease peaked a number of years later, and then eventually it went away. But then you had an overlap of an epidemic in people. These are people with variant CJD. They acquired a TSE by eating contaminated beef. And this seems to be over now. There are no more cases in the UK. So it's a form of cannibalism, really. Yes?
What did that mean, if we recovered meat? What did that do to stop the spread of disease?
Well, this is related to how the meat is, the meal is prepared for the cows. So they used to, like recover the spinal cord by blowing it out with high pressure air or water, and they said you can't do that anymore because that just contaminates it with prions. So they banned that kind of process to limit it. And they also instituted some processing measures that would inactivate the prions. So this is over, but whenever there's a BSE positive cow somewhere in the world, the meat from that country is banned. Yes?
Has there been like a vertical case
A vertical transmission of a prion. I don't know of any. There might be, but I'm not aware of it. You know of any? I don't know. From mother to child? I don't know of any. Okay, so there's a kind of TSE that's infectious. We've been talking a lot about. There's also sporadic, and this is around 1 million people worldwide, typically 50 to 70 years of age. 65% of all the TSEs is this sporadic form. And here it appears without any warning. There doesn't seem to be any relation to eating any infected meat. You have normal genes. Your PRP genes are normal. And you can transmit it to others. So if you give a transfusion or something, or you give blood or some other product that's not checked properly, you can transfer the infection because now you have pathogenic protein in you. And as I said, that's probably how Kuru was established in New Guinea. A person got sporadic CJ, and then they ate the brain, and that established the infection. So that's sporadic. There's also familial, which is a genetic disease. You have a mutation in your gene that predisposes the protein to misfolding, and you develop the disease. But if you donate organs or corneas or blood products and you don't know you have this, then you can infect others. And that's how some of the transmissions have occurred via these kinds of products from people with a familiar form of the disease. So we have 3 kinds. We have an infectious, a familial, and a sporadic. So the familial is genetic. The sporadic just occurs, and we don't understand the conditions that cause the transmission. We don't understand the conditions that cause the conversion of a normal protein to the pathogenic one in these cases. So what's happening here is we have a normal cellular protein. If you eat infected meat, you get some pathogenic protein that converts your normal protein into the infectious pathogenic form. You may have a familial mutation that predisposes you to misfolding, or you may have some spontaneous misfolding that occurs. This is the sporadic form. So 3 different ways that this altered protein can arise. There is a yes?
In the sporadic form, is it always the case that the protein precedes the mutation, or is it just a sporadic mutation?
So in the sporadic form, there's no mutation. It's a normal protein, and somehow it just begins to misfold. And that causes the disease, and then you can transmit it. Because a misfolded protein, no matter how it's made, is potentially infectious. Okay? The only ones with a change in the protein are the familial forms, which have a change that predisposes it to misfolding without any infection or anything else. Okay? So the good news is there is a species barrier. It's not easy to go from one species to another. So if you put diseased brain from one animal to another, you don't always get disease. For some reason, it's inefficient. Most of the time, to get the best transmission, you have to do it in the same species. And a nice example of how to overcome this, if you make transgenic mice that make the bovine protein, you can then infect mice with the bovine prion. So normally if you take mad cow extracts and put them into mice, the mice don't get sick. But if you give the mice a transgene that produces the bovine protein, then the mice will become infected. So the barrier to transmission is in the sequence of the protein. But apparently you can go from cows to people. That's not a problem. But, you know, from, say, a deer to people, we don't know if that can happen or not. So maybe in some cases we're lucky. These prions have a broad host range. They appear to affect many different kinds of meat-eating mammals. So some prions can overcome the host range, and that's why BSE is a concern, because it does, cow BSE can infect people. There are, interestingly, strains now of the Scrapie protein. There's no genetic information, but it can vary. And there are strains that have different incubation times, brain pathologies, electrophoretic profiles, host range, and these all seem to have a different protein conformation. So these are not mutations. Simply slightly different foldings cause the proteins to act in different ways. And these can evolve, amazingly. These can evolve in cell culture. So you can actually take a prion from an infected animal and put it in cells in culture, and the prion will multiply in those cells. And you can pass the prion from cell to cell as if it were a virus. So you can make cell-adapted populations of prions. You can make brain-adapted prions. You can use drugs like swainsinine to inhibit the production of prions in these cell cultures, and then you can select for variant proteins that are resistant to the drug. Again, this is not a sequence change. You're selecting for slightly differently folded proteins. So the point I want to make here is that prions are like Darwinian, can evolve in a Darwinian way. They're subject to mutation, not really mutation, it's not based in sequence, but conformation, and you can selectively amplify them. It's really amazing. Yes? Are there others? Yes, absolutely. Yeast is full of them. My last slide has got that. So this is just to show you that this is increasing in deer, elk, and moose. This is the only TSC affecting free-ranging wildlife. You can see here the areas where we're finding these, even in New York State, Virginia as well. And so again, as I said, this is a problem for hunters because they have to be really careful. So this slide sort of summarizes how all of this may have arisen. I really like this. The blue is naturally occurring TSE and the red is artificially. So probably all of this began in sheep. We can blame the sheep because they can't defend themselves. It spread to wild animals, contact with sheep or eating sheep, for example. It spread to other animals like mink. Mink, I guess, bother sheep sometimes. They could get infected. Could get into cows. They're on the same farm from sheep. And then, of course, the cows led to BSE. The cows probably led to infections of other species by feed product transmission and also into cats. So all of these TSEs probably originated from cows. We got our variant CJ from cows. And a lot of these people who had variant CJ probably transmitted it to others by transplants of various sorts. And then there were probably some sporadic origins, which are natural in humans, from which iatrogenic spread also occurred. So as I said, prions in the food supply is a problem because of the experience with BSE. And a lot of work is being done to try and protect it from this. We're developing diagnostic tests and screening for drugs that block accumulation of prions. Because at some point, some more is going to get into the food chain and it's going to be a problem. Now, the last thing I want you to know is that not all prions cause disease. They have been identified in other microbes as well, and they don't cause disease. They create protein-based memories, and they are believed to allow organisms to switch between phenotypic states. So you have a cellular prion and a pathogenic prion. But imagine in yeast you have two forms of a protein with two very different functions. And this may help the yeast, for example, to adapt to stressful environments. So in yeast, many prion-like proteins have been identified. And these are proteins which can change conformation. They don't change sequence, but they change conformation, and then they have a new function associated with that change. And here are all the native proteins in yeast. And then in the prion state, this is what they're called, and they do different things. So, for example, in mammals, we've been talking about how this conformational change leads to a disease. But in yeast, these changes allow the yeast to use different sources of material to metabolize, altered carbon source utilization, indiscriminate utilization of nitrogen. So they're beneficial. And so probably the vast majority of prions that exist are good. And it happens to be this one causes a neurological disease.
And the proteins switch back and forth between time.
Yes, yes, it goes back and forth in these yeast cases. Now, in humans, it doesn't seem to happen.
So even though it's not really causing any problems in the yeast, could it be infectious, like spread from one to another, the way prions are in mammals?
I suppose theoretically it could, but it's not been shown.